What is it like being inside an egg? Can you feel your sibling eggs nearby? Do you? Can you try to talk to them? Let them know you're here as well? How do you tell your parents when you're ready to come out? We're so delighted to be joined today by Dr. Gabriella Gall, who's here to tell us all about her research into the world of egg communication, specifically the pukeko. Hi, and welcome back to In the Swarm, where we dive into the minds behind the hive. And today we have yet another special guest who's always been fascinated with animal and plant communication and coordination, Dr. Gabriella Gall, or Chini. Hi!
Hello.
So Chini, please tell us a bit about yourself. What are you doing right now?
So at the moment, I'm a postdoc at the Center for the Advanced Study of Collective Behavior. And I currently study different bird systems and I try to understand how first individuals communicate, but also how their communication, which they use to coordinate group actions, actually develops. So in this case, from egg stage onwards, because I look at birds. And in case you wonder what I did before, I did actually not study birds. Birds is very new to me. I did my master's and my PhD on meerkats. In my master's, I was more looking at the group itself and how the group uses territories and how these territories shift over time. In my PhD, it was really more looking at within the group, how they coordinate actions, make decisions to, for example, go back to the burrow in the evening, or how they kind of maintain cohesion while foraging, and how they vocalize and use these vocalizations to do these kind of decisions or make them. And then was a bit of a kind of crazy time trying to find a nice postdoc in a position. So I did some different things. One of them was a very brief postdoc on plant coordination to really understand how, for example, sunflowers coordinate via shade avoidance. Well, I don't know what would come out because COVID happened, and then, well, I mean, you know the rest, I guess. So now I'm here, and it's really nice.
Yeah, now you're here. So you've really mentioned different animal/plant models that you have looked into, but I actually want to take it back even further. How did you get into academia? How did you get interested in this topic of animal/plant communication and coordination?
I guess like a lot of other people, when I was young, I just watched way too many documentaries and they were fascinating. And I was like, oh, I want to do that. I always found biology interesting and behavior specifically. We didn't have it. So I did my bachelor's, actually I didn't mention it before, in Munich. So a university that in itself doesn't actually have animal behavior. Like it was a module part, but very limited. And then I went to Zurich to actually do animal behavior because that's like a major field they have in their behavior biology department. So I don't know. I just was always interested in animal behavior. It's like, this is cool. I want to walk around and observe stuff and study it.
Yeah. And then meerkats. So then from meerkats to sunflowers. And from sunflowers, now we are talking about birds. And by birds, I think you quickly told me behind the mic that you did domesticated chickens. And then you moved on to pukekos.
So originally, actually, the idea was not even to study chicken, but to study pheasants.
Ah, yes.
And, well, the problem with the idea with the pheasants was that you could rear them in the lab and then release them, which is something they do a lot in the UK for like their shoots. And there's a group in Exeter that uses that system to then study, for example, cognition, of how cognition develops and depending on different things they might learn while they're in the rearing stage and then have to use when they're released. And I was like, this is great to look at communication and how that develops over time. And then COVID happened, was still actually ongoing. Brexit happened and avian flu is in full progress. So basically a lot of the shoots that my collaborator had closed and nothing is happening.
No.
And at the time when I started, it was only the Brexit COVID stage where it was already problematic to actually get pheasants and do stuff. And a lot of shoots had already been like closing or like it was very unclear. So then we decided, well, we can't really do pheasants at the moment. And there's a lot of embargoes of what you can do and who can go to university and do things. So we thought we could do a pilot study with domestic chicken because they're basically very readily available. Like it's very easy to get them. And he had all the permits in place for birds and game birds, but also domestic chicken kind of were included. So yeah, then we basically did a pilot study. The problem was, again, because it was a COVID situation, we didn't have a lot of lab space. And so the lab space we had was quite limited. However, it then led to actually finding interesting behaviors that we didn't anticipate that came out of not having the perfect setup for our experiments.
Tell us about that.
So basically what happened in the experiment, we reared eggs, and in the last week of incubation of these eggs, we played them maternal calls to half the eggs and not the other half. Ideally, you would have another control where you would play just a different sound that is not the maternal calls.
Mm-hmm.
But again, we were space and time limited. So we were like, okay, well, we'll do with the 2 bits. And then the idea was to then run experiments in a test arena where we just see how chicks respond to different playbacks after hatching.
Mm-hmm.
And maybe they're more prone to respond to maternal calls if they heard them in the egg, or just be generally more responsive, more vocal, maybe more social.
Mm-hmm.
And so we basically had the test arena where we could do playback experiments. And then we had the home enclosures where we wanted to observe the general behavior. So we had kind of enclosures of, I think, 2 meters by 1.5 meters, something like that. And above them, we had Pi cameras that would record for an hour in the morning and then time-lapse images in the afternoon. The problem was that we were space constrained. So all these enclosures, rather than being very separate and you could go to each one separately, were so close to each other that I had to basically have the walls of the enclosures be low enough so that I could climb over them to do the husbandry for the birds.
Right.
What I didn't know, because obviously I don't know anything about birds, was that chicken, when they're about 2 weeks old, very good at fly hopping.
Oh, yeah.
And so they fly hopped from one enclosure to the other enclosure. And the cool thing, however, is that the birds that did that were mostly birds that had heard a playback in the egg.
Oh,
I don't know why that is.
Oh, wow.
But that's really cool. But basically, had I had more space for my experiment, we would never have found that, because that's not something I would have anticipated happening.
Mm-hmm.
But yeah, they were definitely more exploratory in that kind of sense, that they were just jumping more into other enclosures.
Jumping the fence, so to speak. Yeah, and that's kind of the question that you carry over to the pukekos, right?
Yeah, to some extent.
Mm-hmm.
Yeah, so basically the problem with the chicken is why I didn't look for the pukeko is that the chicken are in a lab.
Mm-hmm.
And then the other problem is that they're domesticated. So a lot of the things that I find, I don't know, is that because they're domesticated?
Mm-hmm.
Would I find it otherwise? And then you get all these comments of like, oh, but this is this specific breed. What if it is a different breed? And you're like, well, I mean, it shouldn't actually matter that much, but you would have to test all of them. It's crazy.
All right. Yeah.
So then the other thing with the lab comes that, for example, when you incubate eggs in an incubator, there's a lot of information that you're missing. And I feel like one very important cue that chicks would have in the egg is the mother.
Mm-hmm.
That obviously will, like, there's a lot of information that she might give indirectly, maybe not on purpose, but that they can use, because obviously they're grasping any bit of information they possibly can, because it's really important for them, for their survival. So yeah, so basically I looked for a system where I could do a lot of the experiments I'm interested in, ideally already in the wild from the egg stage onwards, rather than in the lab and then releasing.
Mm-hmm.
And so that's how I came across the pukeko. So this is a kind of swamp hen in New Zealand.
Mm-hmm.
They're mostly terrestrial, although they can fly. So it's a kind of big rail species. Like if you know how a moorhen looks like, they're basically the same thing, just bigger. The size of a duck, basically. But belong like the proportions of a moorhen in duck size. And the cool thing about them is that they have a really, really interesting social system whereby they can be pair living or pair nesting, but they can also nest in groups that are stable all year round. And when they are nesting in groups, they can also have joint nesting, which means that multiple females lay eggs in the same nest.
Oh, wow.
And that obviously increases or like changes the dynamic between the eggs before hatching of how to coordinate and also the pressures. They're usually precocial birds, so the parents leave the nest with the chicks. And if you're the last one left, well, I mean, there's not much you can do about it. You might be eaten or die because it's cold. But the problem is also if you're too early, obviously you have a yolk sac as a bird that feeds you maybe for another day or two. But after that, if the parent is still sitting on a nest waiting for the rest of the clutch to hatch, you might still die. So that's why there's a pressure from both sides to kind of synchronize. Anyway, so in the pukeko, because you have multiple females laying eggs, there's a pressure between the females of who wants to be the first chicks to hatch, because they are in this in-between stage of not really being fully synchronous. So the first chick that hatches and grows usually grows so much faster and so much better than the other chicks.
Because they get more food.
They basically get more food and just grow better. And so there's pressure that you want, like the parents want, the mothers want their chick to be the first one. The chicks want to be the first one. And so I think this is a very interesting dynamic that's happening.
Okay, and that's kind of hard, right? Because as you mentioned, some of them actually have this kind of collective laying of eggs, and then I guess I'm having this like mental imagery in my head that, you know, you have one big nest, all the mother pukekos actually lay in this one nest, and then they kind of take turns.
Yeah, basically the whole group can incubate.
Okay. Oh,
so
they all sit
males
together.
Well, no, like they take turns,
Uh-huh.
but the males, for example, also incubate, and the females incubate.
Ah, wow.
So these chicks, they're not fully precocial. So once they hatch, they still get fed by birds in the group.
Mm-hmm.
And then at nighttime, they're also being incubated still in like a separate chick hatching, like hatched chicks nest.
Ah, okay.
To keep
okay,
them
so
warm.
once you've hatched, you kind of get moved somewhere else.
Yeah.
Or to the side, and then you get incubated like differently or by a different male, female pukeko.
Yeah, it seems to be the case that they do that.
Okay. Is there any like known ratio in terms of like, because you did mention that some pukeko females will also just lay and hatch her own eggs as opposed to having this kind of relationship, like duality with another female pukeko. Like, is there any statistics for that?
So from what I understand from the literature, is that it seems to be only the really, really big birds in perfect condition that manage to have a territory just as a pair.
Ah, okay.
It all seems to hinge on territory defense, where if you can't defend your territory, you have to allow other birds in, usually males first, to then help you defend your territory. So if you're like a really perfect condition, massively aggressive pair of birds, and you're, for example, in a corner where you have just one tiny edge of territory to defend because maybe the rest is all water, I mean, they're swamp hens, then probably you're great. You're set up. You can do your own thing as a pair, which would be the ideal scenario. But most other birds just can't afford it. So they have to let in other birds to do so.
How did you end up in New Zealand in the first place?
So yeah, basically that happened because I was like, okay, what bird system could I study that has eggs? Then I kind of came across the pukeko and I reached out to, I think it was the former PhD student on the project because he had written a lot of papers. And he put me in touch with his former professor who's actually still working on the system, Professor James Quinn. He's at McMaster University in Canada. And he was saying that they were going back to New Zealand to do a rebanding of the population because it was after COVID that no one had been allowed to New Zealand. And so basically it was the first time they would go and they were like, well, come along. So
you showed up in New Zealand with, I am guessing, a ton of microphones. And how did you get started?
I had microphones. I had tiny, so basically these are Russian spy microphones that we use also on meerkats. So that's what we used them originally for. On meerkat collars to record what the meerkat is calling and with the GPS where it is in the group.
I'm sorry, but why are they Russian spy microphones?
Because they're from Russia. Like there was this Russian company that we can't access anymore because of obvious reasons. And they're basically spy bugs. But the thing why they're useful is because they're being a spy bug. They're very, very small. Yeah. And these specific microphones are especially cool because you can also program them of like the times to record when you want it to record in the daytime. Or like you can have a kind of recording schedule, which for like what I did in New Zealand is not so important. Okay. For what we did on meerkats is very important because usually the microphone battery doesn't last that long. And the important thing is they're really small and light. So basically I used these microphones and I just wrapped them in parafilm and dropped them in the nest of pukeko when we found one that seemed suitable, about 2 centimeters underneath the eggs, so that it would hopefully record whatever the eggs would communicate if they did, and then also the adults that are on the nest to just see the interaction of what's happening.
So the eggs have already been laid, right?
Yeah. So in this case, the eggs had been laid, and I tried to kind of get nests at different stages of incubation. So kind of more early on, in the middle, and then just before hatching, obviously is the most interesting period of time because that's when the chicks can actually really vocalize. The only information that we have on sound of pukeko is from like the 80s. Or 90s. Wow. Okay. And so the data, like the tape recordings, not available anymore. And then the spectrograms in the published paper also, I mean, it's been a while, so they're kind of pixelated. It's difficult to understand what's going on. And obviously you want your own recordings to kind of match it.
So by now we have established that you've been to many field sites. Can you do a bit of a comparison?
So, I mean, the main field sites I've been to, I think, is the one in the Kalahari. So this is the Kalahari Research Center.
For the meerkats?
That's the meerkats. There's a couple of other study systems that are being studied there. So at the moment, still they're the Maryland mole rats, although that project is now closing. They're currently studying yellow mongoose. They're also studying Cape ground squirrels. And then a couple of bird species like the fork-tailed drongo. I think they have the yellow-billed hornbill that they're still studying, and also the pied babblers. The meerkats, I think, are the main kind of flagship species on that side. The site is awesome. It's really, really cool. It's difficult to reach. So there's a dirt road that you have to take from Uppington. And usually researchers or volunteers will come into Uppington and then are picked up by a town trip and then brought to the field site. And the town trip basically already tells you that, yeah, it's difficult to go shopping in the Kalahari Research Centers.
Right.
And the field site itself, because it is a farm, it's also nice because on one hand it's sad because you don't have the big five and you don't have lions and leopards and elephants. On the other hand, it really allows you to walk around and follow your species without being afraid that you might be eaten by something. And so it's like the only site I know where you can really just walk around as you wish at any time of the day.
That you don't have to be escorted.
You don't have to be escorted. You just walk around.
Perfect.
And usually when I first came, people, like the volunteers were there for like a year. And also by now there are more South Africans compared to Europeans because the project was started by Cambridge and Zurich. So there were a lot of students coming from Cambridge or maybe also Zurich. And now it's really nice mixed South African, European. So it's a really nice place to actually meet people from South Africa, because otherwise you would come there and then be stuck there.
Yeah, be isolated.
And kind of be isolated in this farm. For me, it was always nice because I was not there that long. I was there maximum 5 months, which is perfect time. And then you kind of like, you know what? I'm leaving. I'm going back and analyzing my data. So for me, it was perfect. It was really nice. So that's one field site I've been to. Then the last field site I've been to was the one in New Zealand.
Yeah.
And that's very different. In New Zealand, when I was there, was the professor from Canada, his PhD student and me. But at the same time, we were in this, it's basically an open sanctuary where there are rangers and they kind of look after the place, and there's some sheep that they're kind of looking after because it's also like a model farm in a way. And people from Auckland come to on a weekend to just hang out and go for a wander. So it's this very different situation where it's not just this research reserve, but there's just a lot of just people that work there or just are on holiday.
Yeah.
And the other thing was that we were in the, like basically we lived in the hut where the rangers work from. So the rangers, they were there every day. So there were 3 people usually. And they would also tell you all these things of like where the taka here are that you would never see, or like the veta that they release. Do you want to join to release them? And you're like, of course. So these kind of things you would get roped into. And then the next town is 20 minutes away by car. And being New Zealand, that has very poor public transport because there's too many volcanoes to maintain it. Yeah, you have a car. So if you feel like going to the movies, you're like, sure, let's go to the movies. Or we could do, for example, weekend trips to see parts of New Zealand. And we're like, let's go and see a gannet colony on the, I don't know, west coast. And I'm like, sure, why not? So that's very different, but also really, really nice.
So I want to kind of take it back a little bit to the concept of egg communication, because I think maybe some of the listeners might share the same sentiment. I didn't think of chicks or any kind of baby animals still like chilling out inside of an egg to actually be proactive in terms of communication. So my question to you is, how did you first get introduced to the idea? Because you first introduced the idea to me and my brain kind of exploded and I was so excited by it. And how did that happen for you, I guess?
So that happened when I was first kind of looking into doing a study on pheasants, because it had this possibility to rear them from young age and then releasing them. And because I knew I wanted to look at communication, the first thing I did was look at the literature. Okay, what is already published on communication in pheasants, so that, for example, I can decide, okay, what calls am I interested in? And I found exactly 3 publications on pheasants, vocalizations. One was just a very general qualitative description of the repertoire. And then there were 2 that were looking at egg communication. And that blew my mind. I was like, wait, what? And then from then, basically, I was just like, okay, this is amazing. And then just looked into all the egg communication that is out there in this. Basically, it seems that there was a lot of interest in it in around the 60s across different species. Like there's quail hatching synchronization. there's people studied vocalizations in chicken and was really trying to understand this hatching synchronization aspect.
Mm-hmm.
And also obviously how chicks in the egg might communicate with the parents. And then somehow it died down again in the 80s, 90s and then onwards. And so I think now slowly it's picking up again.
Mm-hmm.
But yeah, it's wild. Like for me, it's completely mind blowing, and there's stuff happening where I'm like, this makes like what? I don't know. It's yeah. And also it's really across species. So for example, there are studies, maybe more recent studies actually on like crocodilians of how they communicate to actually call the mother to dig them out, because otherwise they can't even hatch properly because they're in the sand.
Oh my gosh, yeah.
And for the vibration is another one that people look at, for example, because obviously when you're hatching, a lot of what you do as a bird is obviously sound, but a lot of you do is actually movement that then might also kind of jiggle other eggs and give them an indication of, hey, you're hatching. Maybe I should do something about it. And also basically what now is also starting again is trying to really understand what the effect of stimulation in the egg is on the post-hatching behavior. So basically what do you learn from before hatching?
Yeah.
That then is really important afterwards. And there's really cool studies on zebra finches where they, for example, found that zebra finches, when it's really hot, have a kind of called a heat call, which is some sort of panting that they do. And
I see.
when they do that to incubate, like during incubation, it basically already primes the chicks to then be more adapted to really hot and dry environments after hatching, which is really cool.
Oh my gosh, okay.
And so now I have a collaborator, and they kind of, I think they're testing something where that sound itself seems to potentially have an effect on other bird species as well, even though they might not produce that. And this is kind of the acoustic developmental programming, which is now kind of happening that people are studying.
Okay, so we have talked quite extensively about egg vocalizations. And I think one of the main things that got you interested in egg vocalizations is the kind of the behavior that comes after the hatching period, but also the hatching in and of itself, which then gets defined as hatching synchronization. Can you tell us more about that?
Yeah, so basically hatching synchronization supposedly is when you have multiple or like all the chicks in a clutch hatching within X amount of time. However, the X in this case is for me a bit unclear from the literature. So I tried to find an exact definition of hatching synchronization and I couldn't find it. And what I could find is that from my impression, it's defined not over like that they hatch in a specific time frame, but basically when incubation starts from the parents. Because obviously the amount of incubation an egg gets has a huge influence on when it will hatch. And so it seems that if the eggs are incubated with the last egg that is laid, and only then most likely the species is a synchronous hatcher, and otherwise not.
Okay, so it can happen that a bird can start incubating from egg number 1 and meanwhile continues to lay eggs.
Yeah.
That wouldn't result in hatching synchronization. But if a bird lays 5 eggs, and only after it's laid the 5th that it actually goes into incubation mode, then it would be hatching synchronization.
So in an ideal world, you could separate it at that perfectly. And obviously there's probably a lot of birds that really start only with the last egg, like probably quail, for example, that are perfect hatching synchronization. But pukeko, for example, are the ones that start at some point after they laid an egg. It's not quite clear, also because it's a group. So you might have laid an egg and then another female might lay an egg. And obviously they only lay an egg a day. But you might still get multiple eggs on the same day. But then maybe another member of the group, like a male, might be like, you know what? I want to sit on a nest. Why not? And you're like, okay, sure. But so there is a lot of weird variation coming in that is very difficult to quantify. Or like not difficult, but you have to think about how to quantify it.
Well, yeah, exactly. So if anyone's listening and interested in figuring out this big mystery, get into egg research. Okay, so now that I think we've talked a lot about your life in the field, your life and interests as a researcher, and now you are sitting in beautiful Konstanz, no longer in New Zealand, and you are sat with really quite a lot of acoustics data. How's your experience with that right now?
So acoustics data is on one hand absolutely amazing. And on the other hand, it's extremely tedious because basically you have to annotate most of it currently manually, especially if there is no proper vocal repertoire. Like there is a vocal repertoire for Pukeko, but it's so old that it's very difficult to distinguish, okay, what are the vocalizations? And there's currently no expert. So I have to become the expert that now knows all the vocalizations. So basically it means that you have to take at least some of the sound files that you recorded and really go through them with taking the time. And for like a couple of minutes, you might take an hour, depending, and just annotate every single sound that you see that might be a call and give it a name potentially, and then check, and then potentially did a mistake, so you have to go back. And then ideally, we now have a lot of people doing machine learning and deep learning and trying to find algorithms to do these things automatically. But of course, they still need some annotated data that they can use to train the models.
Exactly.
And so even then, you still need to have that annotation. And so, yeah, that's what I've been doing most of the past year, I would say, with some help of some student assistants that also are annotating for me. Yeah, figuring out what these birds are calling. And in my case, while usually you might annotate full vocalizations, so for example, squawk might be your vocalization, I do sound elements. And these elements in my case are defined as like basically structural, like parts of the sound that are structurally very different from other parts of the sound, and or might have like a silent gap in between. The idea being that you might find these different sound elements across different vocalizations, and they might be combined in different ways, which is something that in the Pukeko we can really find a lot of. Like they really seem to combine different sound elements in different parts of the vocalizations and bring them. And it's really interesting. So it's currently more like a linguistic study that I'm doing in a way, rather than a behavioral one.
It's a bit like solving a cipher, right? That kind of reminds me of like what you say, that you need to find the repeated like vocalizations in different places. But I don't even know how I would even wrap my head around this framework with bird calls.
It takes a while. Like basically it was a lot of just starting to label what you see and then go back, okay, is that still the same thing that I labeled at the start? And then you go through and at some point find something new and like, okay, I have to define something new because this is definitely something different. And then you do that a couple of times because it's something you like kind of have done it for a couple of hours and you go back and, oh, this is the same thing as at the start. And then just kind of, you drift slowly over time. I mean, it's really interesting because you really listen in my case, to this nest. The difficulty that obviously I have in my recordings is that I don't have individuals. I have a whole nest. So
right.
I mean, most of the time, it's very easy to see that only a single individual is calling, but I obviously don't know who the individual is. So it might be different individuals calling at a time. So any variation that I might find within nest, yeah, it's just a nest variation. On the other hand, I have lots of different nests, so I can still do a comparison whether different nests uses different, let's say, dialect or something. But yeah, so it's interesting because also having done a lot of sound annotation with meerkats before, where we really record the full vocalizations, rather than the sound elements. I think even if I would do the sound elements for meerkats, I would not find the variation that I find for the Pukeko.
Oh, wow. How is that?
I don't know. They just have more distinct calls, I think, that they used, right? That are not put together by different elements. While the Pukeko or the birds, seemingly more in general, they really kind of, they mix and match. They go crazy. And then the thing is, what they also do is really interesting when they have these crow vocalizations, which is more like a territorial call, defense call, that they get really excited over time, it seems. And then they just add elements. And then the element that they also add becomes potentially more high-pitched and more noisy. So they exchange some elements, make them more exaggerated, and then add some additional elements in the end. So it becomes longer and longer and more elaborate over time. Yeah, so obviously you're still in the thick of it. You're still annotating your life away, more or less. But can you tell us, like, what are kind of the main things or even findings or even maybe the hypotheses that you want to test with your current like data set? Is there anything like that? So the first thing I'm currently doing is really to get this understanding of how they combine these sounds to larger structures. So to the calls, but then also how they combine the calls to maybe call combinations and how they use them. And then the other thing I would be quite interested in is that we have these external cues. For example, we have paddocks where the nests were, where there were sheep, and then the sheep got moved to another paddock. So you have days when we recorded there were sheep there. We have days in the same nest there were no sheep there. And the sheep are loud. They make a lot of noise. They mat a lot. And the same with cows, for example. And then we had a construction site. So there's these external cues and sounds that we might see that might affect how the birds behave. And these are kind of questions, that's the things I can answer with the data that I have.
Mm-hmm.
And anything else, I really, really need to just go and collect more data or like get more context, because that's the part I'm missing.
Yeah.
And then the thing I would be really interested in is to see how, like whether for example, the southern population, do they sound the same? Because they have a very different social system. Maybe they're exactly the same, maybe they're not. There might be calls that I didn't record because I was there just at the start of the breeding season in winter, but I missed the mating part. So that might be missing. I'm missing the summer. So there's a lot of sounds I probably don't have in my recording. And it would be really nice to see, like, and that's just the communication part. Obviously, all the coordination of what do the birds actually do is there as well. Yeah, there's a lot to do.
Yeah, it's still to come, right? So if there's any follow-up, please come back. Come back and tell us all about it. Okay, so now that we're nearing the end of our podcast, I will give you our classic ending question, which is what I like to call pep talk for academics. So if you have any advice for someone who's just starting out or someone who's just thinking about getting into science, because I've watched Discovery Channel all through my entire childhood, preteen, teenage, adult life, any kind of advice you want to give to someone considering a research/academic career path, or any kind of advice you want to give to a PhD, a master's student considering, okay, now that I've done my thesis, do I want to go further?
So I think if you're just about to start and you considering doing research, I think it's important to know that it's just a lot of work to be, because basically I think the main issue is that the scientific positions are not fixed, permanent. Like there's very few of them, especially in Germany. We don't have a lot of the lectureships and things that other countries might have. And so a lot of the time is actually spent applying for positions, which is obviously not what most people are interested in doing. Yeah. And also I watched a lot of documentaries and I'm fascinated by animal behavior and I'm now doing it. But of course, the part where I'm actually in the field collecting data is very little for me, because when I collect data, I get so much of it so quickly that I spend very little time in the field. But that's the most fun part. So if you don't like sitting in front of a computer, maybe it's not the best thing for you. Like I think as a scientist, like it's not like often not shown of how much we just sit in front of a computer analyzing data. A lot of what we do, we could be a data scientist, basically.
Yep.
With just the added benefit of we walk to the field, and with the negative part of we don't have a fixed contract, by the way. It's also, I think, from my perspective, I really like science because of the people, because you meet a lot of amazing people that are super fascinated, motivated, interested. Yeah, and also the thing, I feel like the problem with the academic system, I mean, it's a problem and it's not, but it's built in a way that basically the higher you go, the more you actually are supervising or mentoring. So if you really take joy from mentoring, that's obviously great. Or like if you just have so many ideas that you know you can't deal with them yourself because there's just too many of them, perfect. If you're like, I really want to do the analysis myself because this is the part I super enjoy, maybe it's not the best place. So yeah, there's like a lot of things to weigh. For myself, I don't know, as long as I find the funding, I'm like, this is great. Once they don't fund me, I think something else will also be great. Yeah. That's the thing, like, don't put yourself down. I think you should never feel, if you're in science, that, oh my God, you're like, this is the only thing you can do and everything else is terrible. I don't think that's true, because there's a lot of amazing people that actually do really cool research that are not in science or like in academia. Yeah. And there's so many, like, if you can get fascinated for like random stuff in biology, it will still be there. I mean, you can still get fascinated with it just from a different perspective, which is great.
Perfect. Thank you so much for your time today. And I really hope that we get to hear more about egg communication and vocalizations and behavior, collaboration, cooperation, and everything else that you'll find out from your future research. Thank you so much.
Thank you.
Yeah.
This was really fun.
And this has been In the Swarm. Stay tuned for next time. Bye.